Right, shall I go ahead and share my screen next? Ramona, if you stop sharing, and I will see if I can get mine up and running. And hopefully, right, I hope everybody can see my screen. Thanks very much, Ramona, for a great talk. And I'd like to carry on the theme. But before I go any further, I'd like to acknowledge two outstanding groups of people. The first are the scientific and clinical colleagues whom I work with as part of the radiomics team at the Royal Marsden Hospital and Institute of Cancer Research. And the second are the staff of the NCITA Repository Unit and its chair, Avis Sala. Avis was very disappointed due to unforeseen circumstances that she couldn't deliver the talk that she'd also originally planned for this session. But we're really grateful to Ramona for having given us such a great talk there. Now, NCITA is one of the sponsoring organisations for this year's workshop, and I'll explain a little more later about who we are and why we're interested in XNAT. So you've just seen from Ramona some particular applications of XNAT in the cancer domain. What I want to do now is move the discussion on to think about the challenges associated with data more generally in cancer imaging and how XNAT helps us to address these. The talk's going to move backwards and forwards between the three themes shown here. And so the Institute of Cancer Research, the organisation for which I work, is particularly interested in translational research. So this slogan, from bench to bedside and back again, that I'm showing here, describes very well the sort of ebbs and flows of the projects that I'm involved in. So we can divide imaging data for cancer research into three broad categories that reflect this: experimental research, clinical trials, and projects involving standard of care clinical data. So I wanted to start by looking at some of the characteristics of these imaging data in relation to the needs of AI algorithm development. And it seems to be that AI is currently the trend in radiology development that's sweeping just about everything before it. So without even reading the contents of the table, you can see from the colours that our three categories of data are very different in terms of their suitability for algorithm development, but none of them are ideal really. Probably the most crucial factor is how many imaging data sets are available, because that's what's going to determine firstly whether deep learning is even feasible, and after that the generalizability and any biases that algorithms might have that we develop. So here, routine healthcare data is probably where we need to look to accrue patients, particularly in rarer cancers. However, as sort of illustrated by all the red entries below that, standard of care images might be unsuitable for developing AI models of cancer for other reasons. They have variable quality, they tend to be very poorly controlled in terms of protocol standardization, and they usually lack the level of detailed annotation that comes with advanced clinical trials. I work mostly in what might be called the sweet spot of clinical trials and clinical studies, and these often have high quality quantitative data, and that can potentially be transformative in terms of predictive power. But these are expensive to obtain, and it really needs quite a lot of effort on behalf of the clinical physicists involved, for example, to harmonise acquisitions between institutions in multi-centre trials. And that inevitably leads to smaller numbers. At the other end of the scale completely, experimental studies of new techniques can be really powerful in terms of the richness of data for individual subjects, but many of the variables that we collect might not be realizable in standard of care diagnosis, and so probably aren't suitable, at least in the near term, for algorithm development. However, almost inevitably, future cancer research is going to involve all three categories of data, and as can be seen from the posters in the workshop here, XNAT's being developed very actively in all of these areas. A research repository makes it easy to store, browse, and access data both manually and programmatically in a way that's open, vendor neutral, and extensible. So we've already heard quite a lot about DICOM data repositories, but increasingly high-impact team science involves much more than just clinical imaging. So with a research repository like XNAT, we can aggregate data of arbitrary types, including non-DICOM data modalities, and this is something that you just wouldn't fit into your standard packs. And then what we clearly want to be able to do is to process those data in original and potentially arbitrary ways, perform the automatic segmentation we've heard about, perform modeling and analysis. And then, although this is still very much a work in progress and varies between different studies, our aim, after we've processed the data, is to store the results back into XNAT together with their provenance, allowing us to audit studies and reproduce work. And then finally, what we want to be able to do is use any search item in the image metadata, and eventually probably to use machine learning itself to perform content-based image searching without humans having to do laborious tagging of imaging. And all of these resources and the enhanced visualization that goes with them is then going to be brought together to allow clinicians and scientists to participate in virtual tumor boards, taking advantage of the experience wherever it might be in the world. So our ability to spin up multiple XNATs as and when we need makes it very easy to establish an environment where it's easy to manage the flow of information in our research. Depending on the information governance framework for any individual project that we might be involved in, we can locate our research data either on a server within the National Health Service or within our academic institution, or potentially make it more widely available for things like Kaggle challenges and so on. Our major funder for all this work is Cancer Research UK, which is a charity. And at the end of 2019, it established the National Cancer Imaging Translational Accelerator, or NCITA for short, as an investment in infrastructure across 7 of the leading UK universities. Now that's since been expanded to 9. So our focus is on delivering trials and clinical studies that involve specifically advanced imaging. So NCITA is built on 3 core units: a QA/QC unit, a trials unit, and an imaging repository unit, which is what I manage. That repository unit uses XNAT to facilitate multi-center trials, and the units are supported by contract management, engagement, and training groups. So, although we operate on a rather smaller scale than exists in the US, we cover many of the same areas as Quinn, IDC, and TCIA, all rolled into one, at the same time as providing the sort of procedural and statistical expertise of a traditional trials unit. So this combination of experience is pretty much unique in the UK. When I started the job, it rapidly became clear that despite a keen desire to collaborate, the partners weren't in favour of a single large-scale repository located at any one of the individual institutions. Both for political and information governance reasons, our thinking moved rapidly towards the idea of a federated discussion in which all of the XNATs are peers rather than some form of hub and spoke model where there's one super XNAT governing everything. My colleague Chris Rookyard has posted demonstrating our first technical steps in achieving this, and I'm really looking forward to the town hall session on XNAT Federation tomorrow. So we want to make it easy to access all the different hierarchical levels of those data, all the way from the final results of a set of trials, going back to even tracing all back to an individual voxel if necessary. Well, why is that so important? It's because each level can tell us different things. So let's think about that video, still from that video that we saw earlier. So these are separate metastases that have been segmented, and what we've done is a diffusion-weighted imaging scan, which has produced some histograms. So these histograms have apparent diffusion coefficient on the bottom for all the different metastases. So the histograms are all different, but experience tells us that their shape and the value of ADC at the peak can help us distinguish between the different types of lesion. So if we then choose to analyze not just the simple statistical features of the data, but also other properties like image texture, and maybe couple that with some genomic data, very much as Ramona was talking about, then the analysis might help us both with diagnosis and treatment decisions. If we then step up a level in the hierarchy and consider all the lesions for a patient, we may be able to arrive at a prognosis. So here's an example of some lovely work by Matt Blackledge and colleagues at the ICR, where a simple threshold on the volume of lesions was found to be a very strong predictor of survival. Stepping up yet another level, we now add time as a dimension to the analysis. And this data from Dave Collins and Matt Blackledge shows the results of serial imaging on a 49-year-old woman with ovarian cancer and 4 large mets. So quantitative analysis of the sort that we saw earlier shows that there are 2 distinct classes here coded as red and green. But what happens when we add the chemotherapy treatment? So those metastases evolve over time, those lesions evolve, and you can see that the diffusion coefficients here rise and the diffusion coefficients in the green ones go down. And following all these things is something that we can do very easily when we have all the data in one place. Then finally, looking at our levels 8 and 9, we can see that results from a single trial might tell us about efficacy of a therapy, while accumulating all the data into a single database could make it really easy to perform a meta-analysis of the studies and audit the entire process. So this is the sort of thing that we'd really love to be able to enable. So, what are the key features of XNAT that allow us to do these sorts of things? Well, this may seem really, really basic and really simple, but role-based access with data organized according to projects that depend on our academic idea of what a project might be, it's really, really important. And PACS generally doesn't do this for images. You can retrieve the data and metadata from anywhere in the world using standard methods like the REST API. And now shortly the XNAT team will be introducing DICOM Web as well. You can store arbitrary data. You can augment the types of data that XNAT knows about natively with schema extensions. You can use the Manage Files option to store a whole host of supplementary data relevant to the study that you're doing. Then the extensible functionality of XNAT also includes plugins, a container service, and a whole host of whole ecosystem of associated tools. And I'd like to say at this point, what's also great is to have such a responsive and collaborative core team at WashU with whom we can kick ideas backwards and forwards for new tools. So when I formulated the strategy for the NCITA Repository Unit, the first thing that I wanted to focus on was the XNAT OHIF viewer, on the basis that this is the part of the system that our radiology and radiotherapy colleagues will interact with the most. So we've made a lot of basic usability improvements, particularly in the area of how we handle regions of interest. So here's the example of imported DICOM RT structs directly from the radiotherapy planning system. Ramona and Lorena have already talked quite a lot about the NVIDIA AI annotation. This comes really as an it's something that is an experimental feature in the latest released version of the XNAT OHIF viewer integration, which comes as an XNAT plugin. But you have to add in an AI AA server to be the thing that XNAT and OHIF communicate with. And there's a nice video in the new training course that we're launching that steps you through exactly the procedures you need to install that NVIDIA server. We've also added numerous additional tools for manual refinement of these AIAA results, but there's a lot more work to be done to combine these tools with more comprehensive electronic case report forms, and we'll be talking about this more in the town hall session tomorrow. Now, coming back to the question on digital pathology, we're really excited that DICOM Web will soon be available to XNAT. It's been great to work on the pre-release version with Dan's team. What you're now seeing is a sneak preview of one of the early wins from this. Sharp-eyed observers of the XNAT discussion forum will have noticed that we've now enabled DICOM, GM and SM IODs as session types in XNAT, and we're currently on an alpha build of our new integration of the whole slide viewer with XNAT. And you'll have seen how we can just click through from one of those new XNAT session pages to interact with the microscopy data in a way that should be completely intuitive. And this will once again extend the range of data types that we can work with in XNAT. So many of our technical developments are closely linked with the delivery of academic studies, and this is a use case that's currently driving our microscopy developments. It's the CIUK Sarcoma Accelerator is a pan-European collaboration to link clinical, digital path and radiology data with genomic studies. So sarcoma is a cancer with multiple different types, some of which really aren't very common, and it would be highly difficult for any individual institution to accumulate enough data to make this type of study feasible. So XNAT is going to provide the main user portal for the whole study, and then within the web app, we'll hand off to CBio portal for the genomics aspects of the study, which we don't currently cover. So I've already mentioned the federation aspects of the repository unit strategy, but there are other key areas that we want to deal with, particularly fair data access. And I just want to give a quick shout out to Steve Moore's poster. And so I recently discovered that there's a really good synergy between the work that the NeuroBridge project is doing and what I want to deliver in conjunction with Health Data UK, who are our national coordinator in this area. So finally, in my last slide, I thought it'd be worth illustrating a complete machine learning project and pointing out just how many steps involve XNAT. So in the overall picture, XNAT, the niche XNAT occupies is this middle research layer that involves getting data from the hospital data sources, anonymizing it, putting the curated data in the repository, and then developing the AI model, and finally deploying it for research partners. And that is all the stuff that is currently really difficult to do in preparation for your workup for CE marking and FDA approval, because you have to get evidence. And so if you think about the steps involved and the tools that XNAT provides, then we can see that all the different tasks involved in preparing in anonymizing the data, preparing the data, performing the modeling, all of those are aided by XNAT. And so we're very happy to be sponsoring the workshop and using XNAT for much of our research. And so thanks very much for the opportunity to talk. And I hope I wasn't on mute for all this time.
Why don't we give Simon a chance to answer a couple of questions, and then we'll take a 10-minute break. I don't think I'll need much time to wrap up at the end, so we're going to shift things up by just a few minutes.
One of the questions was, what is your mechanism for searching across all 7 NICRA's partners? We are looking to build a portal for the Australian Federation with 12 partners. Custom plugin?
Yeah, so okay, so we've just been creating, and and this you'll hear much more about in the poster session. We've been creating a new OpenID plugin for XNAT that allows each XNAT to be effectively a resource provider. So we use a common authentication platform, which could be anything, but we've chosen AWS Cognito for that platform, in which we create a user pool. And users in that platform can be associated with all the XNATs that they have accounts on, and without needing to share any passwords directly from XNAT, we can effectively get simultaneous JSession IDs from all the XNATs that somebody could be logged into, and then run queries on each of those individual XNATs and merge the results and present the user with their data. So hopefully Chris will have a demo of that later on in the poster session.
There are a few more. How are you generally receiving images from sites XNAT, desktop software, disk, other?
So one of the really interesting things about working in multi-center clinical trials is everybody has different access to be able to do things. So some hospitals will not let their sites install new software on machines. So that means that that a lot of people can't install the XNAT desktop client. So often sites will use the very easiest thing, which is the zip compressed uploader. That works just fine, and they'll do their own anonymization separately. Some people might use DICOM browser, for example, to do the sending. Some people might use the XNAT desktop client. So one of the things that we have to do a lot of in NCITA, and we have somebody whose job it is primarily to do this, is to go off and talk to sites, do the due diligence, find out what capabilities their institution will let them use, and then we work out how we're going to do that upload. So a variety of mechanisms is the basic answer.